Okay. It's my pleasure to introduce Dr. Lara Symul to come and talk to us at DBMI. Dr. Symul is currently a postdoc at Stanford, but before that, she did her PhD at EPFL Zurich, and part of that work was combining omics data with mathematical modeling to learn about the regulatory dynamics of circadian clock. And during her postdoc, she has this really cool, area of research where she's analyzing self-tracked phone app data with clinical multiomics to study various aspects of women's health and especially menstrual health. And this is what I'm hoping that we're going to listen to today. And one interesting thing that I also noted was that Laura has this background also in scientific illustration and data visualization. So I'm assuming the slides are also going to be really cool. So welcome, Laura, and we're looking forward to your talk.
Thank you so much. Okay, pressure is on. I hope you like the pictures and visualizations. But thank you so much for the introduction and the opportunity to present some of my work. Indeed, so when I started my postdoc, one of my questions and objectives from a biological perspective was to understand or contribute to understand a little bit what was the impact of frequent hormonal fluctuations on women's health, both physical and mental, and on their well-being. As some of you may know, there are really tight links between the reproductive system and the immune system, just from evolution, from having to survive as a species, right? And when looking at that from statistical perspective, there are several challenges. And indeed, some of my early work, which actually I'm not going to talk so much about today, but had to do with data collected from menstrual cycle tracking apps. And one of the things that you need to deal with in this context is that there is a lot of variability between individuals. Not two women have the same reproductive journey and menstrual cycle. And then, of course, there is a lot of missing data, right? It's voluntary tracking, so you can expect a lot of missing data. And what we found out is, and that's also expected, is that the missingness is not at random. So depending on what's the biological state of the person, then there might be more or less missing data or different variables missing at different times. And so that was one of the challenge in this, and still we were able to recover the biological states from this data, provided that the data quality and data frequency was high enough. And what I'm going to talk about today, though, is more on the omics side, and that has to do with microbiota, the vaginal microbiota, and subcommunities of the vaginal microbiota. And so that includes various type of data, and today I'm mostly going to talk about sequencing data. And before I jump into the methods, I'm just going to give a very quick introduction to what the vaginal microbiota is. And it's simply really the set of bacteria that is found in the vagina. And for anyone who's familiar with the gut microbiota, one thing that is very different is that it's much less diverse than the gut microbiota, and also less diversity is a good thing in the vaginal microbiota. So a vaginal microbiota that's fully dominated by one of the lactobacillus species is considered a healthy microbiota. And on the other hand, when it's diverse, then it is associated with negative health outcomes. And so when there's a lack of lactobacillus, then there is often what is called bacterial vaginosis, and increased risk for preterm birth or susceptibility to sexually transmissible infection. And so there is a need to be able to describe a little bit better the structure of a dysbiotic microbiota. And so just for a little bit of history here, so typically historically the microbiota structure has been defined through clustering approaches and defining different community state types. And that goes back to one of the first study where many microbiotas had been sequenced, and then clustering was applied to that, and 5 clusters were identified. 4 of them were actually dominated by one of the most common lactobacillus species, at least in Northern America. And then the 5th one, which is number 4, is this everything else bucket, right? And so several studies have followed showing that indeed Participants who had an everything else community state type were more likely to deliver preterm. But also what was found is that not all lactobacillus were equal, in the sense that some lactobacillus species, for example, inners, was more likely to be permissive of non-lactobacillus species, right? This is what you see here in this diagram, transition diagram, that the state 3 is more likely to switch to state 4 than the 2 is, right? But so still this everything else bucket is not very satisfactory, because if there are maybe some substructures or some features that are especially associated with negative outcomes, then we wouldn't know because it's all lumped together. So again, there were attempts to use clustering to refine that type. And so this was done some years ago, 3 years ago, about 3 years ago, where that gave 7 sub-CST, sub-community state type, and you see their composition here in shades of red. But what you see is that there is, first of all, there is a lot of redundancy, right, between some of the types. The 4A and 4B are quite similar. And then the 4C is a bit separate. And then here we have C1, C2, C3, C4 that are kind of redundant except for one dominant species. But there are fundamental limitations about clustering when we think about microbiota composition, because in clustering, samples are fully assigned to just one cluster. But in fact, if we think about bacterial communities, then several bacteria, they could coexist and they could live together. So here, this one sample that is not even very close to any of the centroid is fully 100% cluster 1. One other problem with using a clustering approach is that we don't see the transitions between the different types. And as we know, the vaginal microbiota is actually quite dynamic. And so we don't know if a transition from a blue state type to a red state type actually happened very fast, rather slowly, or if some other subcommunities were necessary to actually do the transition. And so there was really clearly a need for a better statistical model. And so the one that we used is a topic model, because this is a mixed membership model. And so on the other, so if we compare with the clustering approach here, so this sample here is not anymore 100% cluster 1. It could be 70% topic 1, 2%, 20% topic 2, and 10% topic 3. But in a sense, topic and clusters they play this role of prototypical samples, right? Prototypical compositions that would correspond to species that often co-occur and so on. So what are topic models? So they were first introduced for population structure, but they really became extremely popular when they were reintroduced as natural language processing models. It's a Bayesian model. And what is observed, and so the reason why it's topic models, because what this model are trying to do is to identify topics in different documents. So the observed variable is the count, the number, the frequency of words in each document. And that is modeled through 2 latent variables. One that is the document composition, saying how many topics there are, or like what is the proportion of each topic in that document. And one that is the beta matrix that is going to tell us what is the composition of those topics. And so just to give an example, and I hope this will resonate in Colorado, if we have a series of documents about running and climbing and outdoor sports, then in these documents, we have a series of words, right? Like race, rock, harness, polar, and so on. And we could identify with this, applying this model to these words and documents, we could identify 2 topics, one that is about climbing, one that is about running. And these 2 topics are defined by differential frequency of words. And so words like boulder, harness, and rock are a lot more likely in the climbing topic, while marathon or race or cardio are more likely in the running topic. And some words could be equally likely in both topics, like shoes. They're both important for climbing and running. And so in the microbiota world, our documents become our samples. And then our words become the bacteria or the ASV if we do 16S sequencing. And then the subcommunities are these topics, right? They're consortia of bacteria that live together, benefit from each other, but could also coexist, right? You can have a magazine that talks both about climbing and running, and so you could have a sample that has both of these topics. And the estimated relative frequencies then would just be the multiplication of these two matrices. And so the great thing is that we can apply this directly to the matrix of the count data. In the context of the vaginal microbiota, we have very sparse matrices. So you see here, and it's a bit unusual, but the features are in the rows and the samples are in the columns. So that tells you how many of this ASV had been sequenced at that sample. And we can fit the LDA directly to these count data. There is no need to transform the data in any ways. And so if we do this, then we do have the beta matrix, the topic composition matrix. And so we see here for each of the 7 topics, what is the proportion of each of these features. And the first thing we can see compared to the clustering approach is that we have much sparser topics compared to the clusters. And then we have the gamma matrix, the sample composition. And so here we see that some of the samples on the x-axis are only composed of one topic right here, topic 5. This sample is mostly topic 5, but then many samples are actually a combination, a mixture of these different topics. Just like clustering, for the topics, we need to choose a number of topics. We need to choose K, right? So how do we do this? How did I pick here that there were 7 topics? So typical approach is to use cross-validation and look, for example, at the perplexity and see if there is any elbow in that. The problem is that when we do this, so on simulated data, that works perfectly. On real data, it often doesn't work that well, just because the data doesn't follow exactly the LDA assumption, right? So that could trigger 2 reactions. So one is to say, okay, let's write a better model so that it matches better the data that we observe. But that could be very difficult to do because we don't necessarily have a good idea of like what is exactly the biology. And then the other thing is that we could still think that we can use a relatively simple model like the LDA, think that there is some usefulness, and try to find post hoc analysis mechanism to identify where there is deviation in the LDA assumption and see what we can learn still. So that was the approach that we took. And the idea that was behind this idea of topic alignment was to say, okay, let's say that there is actually 5 true subcommunities. Then if I fit my model with 7 topics, for example, then 5 of these topics will likely identify those true subcommunities. And the 2 remaining ones are just going to be some shuffling that depends on the data set characteristics of the remaining bacteria, maybe the bacteria that don't really belong to any subcommunities, that are just like part of the background noise. But then, so this was if I fitted with 7 topics. But if I fit with 8 topics, then I'm still expecting to find those 5 topics. And then the 3 remaining ones will be a reshuffling of the remaining bacteria. But then in both cases, in the 7 and in the 8, the 5 topics that identify the true subcommunities, we expect that they will have a lot of similarity, and that the 2 remainings or the 3 remainings will kind of be a reshuffling of the remaining bacteria. And so this is the idea behind topic alignment, is maybe we can identify the true subcommunities versus the spurious ones by looking at the similarity across resolution of the topics that we find. And so the idea is the following. So here we will fit the models at different resolution, and in this example from 1 to 12. And so if I represent this graphically, we have here for 4 topics, we represent these by rectangles, and the height of the rectangle is the overall prevalence of that topic. So it's the sum of the topic proportion over all the samples. And if I fit my model with 5 topics, and connect those topics by similarity or alignment weights, and I'll come back on this, then we see that we have 3 topics that in fact remain almost the same, and then 1 that splits into 2. And if I continue that over the whole resolution, then we have a topic alignment diagram, and we can see that there are some paths that are pretty well conserved throughout resolution, right? This one here, the green one, and other topics that don't really belong to any path, like this one here is likely a spurious topic, right? So one of the key questions is, how do we compute those alignment weights, right? How do we connect the topics together? And there are two ways to look at this, just from the duality of the topic model, right? Remember, we have two latent variables. One is the topic composition, and one is the document composition. And so if I look at two topics or three topics, if I look at the beta matrix or the gamma matrix, I can see which ones are similar from these two perspectives. And so one way to compare them is to look at the topic composition, and the alignment weight is found by solving an optimal transport problem. And the other possibility is to look at the sample composition, and there, oh, what happened? Do you still see my screen? Okay, it disappeared for, okay, sorry, disappeared for me for a moment, but all good. And so if we look at sample composition, then what we proposed was to do the product, the cross product of the topic composition. And so this is why we call it the product alignment. So just with a little more detail. So what we have here, so we have our topics lying on the simplex. And so we have a model with two topics in green, a model with three topics in blue. The height of the topic is the mass of the topics, the height of the rectangle in the alignment earlier. And what we're trying to see is finding the weights here, the pairing weights that are going to minimize the total cost of transporting mass from one model to the other ones. The cost that we used here is just a Jensen-Shannon divergence, and we could have used another metric of the similarity between topic composition. This is the one we used. And then we solved the transport problem for this distance. In the case of the product alignment, then what we do is literally the cross product between the gamma matrix and one resolution with the gamma matrix at the other resolution. And so that will give us that topics that have similar patterns across samples, right? So if one sample is primarily described by topic A1 and topic B1 in another resolution, then we expect those A1 and B1 to be the same. And so if we visualize for the same data, the transport alignment and the product alignment, we have something that's a lot sparser in the transport alignment, and that's just due to the fact that we solved an optimization problem. And in the product alignment, we have something that is a little messier. But in terms of what those alignments tell us, this would be very similar. So here, this is data simulated with 5 topics, true 5 topics. And what we see in both cases, and we see it more clearly in the transport alignment, is that we end up with 5 paths that subsist, and then the other topics, they don't really stay on a path, or they don't really have their own path. And even though it's less clear here, we still see that those 5 paths that subsist after 5 topics. So this is just a visualization, but we also defined some diagnostic scores to help us diagnose that and apply this in a more quantitative way. So one thing is to count the number of paths, and we define paths as the topics that are connected by the highest weight. One score is the coherence score, and that would tell us how similar 2 topics, or not 2 topics, but a topic is within its path, its own path. And so we would see here that, you know, again, topics connected by high weights or have higher coherence scores, and topics that are not well connected have very low coherence scores. And then we also have the refinement score, and this one is a little different, and it tells us a little bit how tree-like the structure of the alignment is. And that is useful because not only actual topics would have high refinement scores, but also in a phenomenon in biology that's called strain switching or functional equivalence. And that is when two bacteria can basically play the same role, but they don't necessarily coexist with each other. So when those phenomena occur, then we would expect to see topics with higher refinement scores. So that also has a biological meaning and interpretation. We did a lot of simulations to see how these scores behave when we deviate from the LDA assumptions, because this is what we expected in the actual data. And so here we added some background noise. So we simulated the data such that the alpha parameters defines how many, like what is the proportion of the bacteria that belongs to subcommunities versus don't belong to any subcommunities. And we see that we have local maximum or like smaller variances in those scores at the optimal number of topics, but that increases, like the noise around that increases as the alpha increase or decrease in this case. And then another phenomenon that can happen is the strain switching. And so there we were also looking for where we see a plateau in the number of paths and whether the data would still correspond to what we expected. And it does. So all of that has been implemented in our package that's called ALTO for alignment of topics, and has a website if anyone is interested in trying it out. So we applied that to actual vaginal microbiota samples. And we had samples from pregnant participants and from non-pregnant participants. And when we applied the topic alignment to those samples, this is the alignment that we find, with an optimal number of topics of 9 topics in that case. And so again, as I mentioned earlier, we have sparser topics than there were clusters and the clustering approach. And if we still compare those to the clusters that had been identified, so what this is showing is the similarity between on the y-axis, these are the clusters that had been identified, and on the x-axis, these are the topics. And so we see that for the lactobacillus dominated topics, so the 1A, 1B, 2, 3, and 5, there is a good match. And for the non-lactobacillus dominated, there is some correspondence, but it's not perfect. So why do we have 2 topics for 2 clusters for 1 topic? Is because in the clustering approach, if you have a little bit, or if you have a majority of Lactobacillus crispatus plus something else, then you kind of need several clusters to be able to reflect those different realities. While with the topic model, you only need 1 topic that has the Lactobacillus crispatus, and then you can add to it any proportion of other topics. So in the clustering approach, you kind of have to multiply the number of clusters to reflect the fact that crispatus can be with iners, or can be with Gardnerella, or can be with something else. So we need fewer topics to obtain the same accurate representation of the samples, just because we can have this mixed membership. Here's some of the difference between the clusters and the topics can be just explained by some taxonomic differences, but not just, right? We saw that the clusters were a lot less sparse, and so we identify cleaner subcommunities with the topics. And then here, what we see with those sub-CST 4C 0, 1, 2, 3, 4 is potentially this phenomenon of functional equivalence, where you have a group of bacteria, a subcommunity, that can coexist with 3 different, or in this case, 4 different species that are mutually exclusive. When you cluster samples like this, you need 3 clusters to describe them. When you use topic models, because it can deal with synonym, then you don't need so many topics. So this is what we find. But one of the main advantage of using topic models is, first, it describes better the transition, but it also predicts much better the transitions. So one of the things that's clinically relevant is whether an individual is going to lose lactobacillus dominance. And here what we see, and so this is a rare event. I'm showing here the F1 score, which is the harmonic means of precision and sensitivity. And so what we see is that topics are much more able to predict when an individual is at risk of losing a healthy microbiota. So that was very interesting from a clinical perspective. So here, what I'm showing is a time series from one of the pregnant participants. So we see from early in pregnancy up to delivery, here a little bit before term, but not too much. And we see the description in terms of sample clusters. And here we see what's under the hood, actually, right? Where we see that there are transitions between the topics, and that some topics increase in proportion and then decrease. And there is not necessarily an abrupt transitions in those subcommunities. And this is showing the same for non-pregnant individuals, where the participants were followed for 10 weeks, and most participants provided samples daily. And that links me back to one of my initial interests on the menstrual cycle effects, right? Because what we see here, so we have actually 2 menstrual cycles for that individuals. This is the bleeding, the menstrual period here. And what you might notice is that there is some sort of repeatability in the microbiota composition, where we see that there is more of topic 1 early in the cycle and more of this topic for BA late in the cycle. And so one of the questions was whether there was any menstrual cycle effects in the topic composition for the participants. And what we found in a cross-sectional analysis is that, so topic 1 decreases, so crispus decreases, and then 2 topics here, the red, the C0 and C1, they increase during menses. And what is very interesting, and this is why I brought up the alignment plot again, is that these 2 topics are actually topics that branch out pretty early. And so in all the non-lactobacillus dominated topics that are in red or oranges or orange shades, we see that these topics that are really increased during menses, they actually branch out pretty early from the other non-lactobacillus topics. And so we were interested in looking whether there was more about the menstrual cycle effects. And so while there were only 2 topics that had cross-sectional effects, when we look at longitudinal effects, we see a stronger correlation. Is there a question? I'm not sure if I'm okay, good. So yeah, what this is showing is that if we look at participants for which we had at least 2 menstrual cycle worth of data, then there is a strong longitudinal correlation between the cycle. And so that is very interesting and somewhat like not well studied. And what is missing here is actually hormonal measurements, right? All we have is the bleeding patterns. And so I think there is some interest from the microbiota community to look at the interactions between the hormonal changes and how bacteria react to that. And sometimes, even if we don't see a strong effect at the topic level, so here, this participants, for example, we don't see strong changes in the topic. But if we look down at the taxa level, then we find again those very strong correlations from one cycle to the next. Some of the future work is also going to investigate deeper the interactions between the cytokines, the metabolites, and the microbiota composition, because we do find that half of the cytokines have elevated, so those are the host cytokines in the vagina. They have elevated concentrations around the menses. This is not too surprising because it is an inflammatory process. And then we also find that about 10% of the metabolites have an increase around the menses or a decrease around the menses too. And so some of the follow-up question will be to better understand the relationship between inflammation, local inflammation, and topic composition or microbiota composition, and also understand a little bit better whether there is a link between inflammation and the risk of preterm birth, because one of the topics that is associated with menses also has some association with preterm birth. This will have to be validated in different cohorts, but this is what we observed in the data that we had. And so this raises new biological and statistical questions. So one is that, okay, if menses topic are associated with preterm birth and menses are a time of elevated inflammation, then is there a link between inflammation and preterm birth? Can we detect that early? And which one is coming first? There is no hope in drawing an actual causal diagram and trying to find out causation in this case, and it's really hard. I mean, a lot of parameters are not in our control, but it still raises interesting questions in terms of what are the redundancies and what are the specificities of each of these modalities, right? Do we see a stronger association between the cytokines and preterm birth or different outcomes? And what is the respective contribution of each of these modalities to the outcomes? So I started to explore a little bit this kind of multi-omic integration and prediction, and where we have different modalities, so the microbiota composition, the cytokine concentration, and we're interested in sometimes composite outcomes, right? It's not necessarily always like, is it preterm or not preterm, but maybe what kind of preterm birth do we have? And because it's a multi-etiology health outcome. And so there we are thinking in terms of multi-omic alignments and just using the word alignment in correspondence to topic alignment that are presented. But there are several multi-omic integration methods that already exist. Some are unsupervised, and I'm thinking about the MOFA or the status methods. Some are supervised, and we can think of sparse PLS or multi-block PLS. But a few of, like almost none of these methods are really also trying to look at the biological knowledge that we already have between some of these modalities, right? There are networks that exist between the microbiota, the bugs that are present in the microbiota and the metabolites that some of these bugs can produce. And so I think there is really exciting work that can be done in using those methods and augmenting them with some priors that we have about the biology and about the relationship between the metabolites and the microbiota composition. And so that's going to be some of the future work that I'm excited to look at. So continuing to understand a little bit better what are the drivers of changes in the microbiota environment, what drives stability or path to recovery in the microbiota. And then obviously one of my plan is to also have long-term longitudinal assessment of that and better understand the relationship between the menstrual cycle and the microbiota, and then see how we can use tools like apps and connected device to also gather clinical data from the individuals. And beyond the vaginal microbiota, I'm pretty convinced now that the data from these apps, under some guarantees or some tracking properties, we can use that to look at the impact of the menstrual cycle on various chronic conditions. And one of them that I will set out to look at more precisely is to look at the direct and or indirect effects of the menstrual cycle on the glycemic response. So from anecdotal evidence, we know that some patients that have diabetes, they have to adjust a little bit their medication throughout the cycle. But in healthy individuals, it has not really been studied whether there were direct or indirect effects on the glycemic response. And that is because it requires really high resolution data for being able to distinguish between those direct effects or these indirect effects, right? Because the regulation of the blood sugar or the blood sugar levels, of course, they primarily are driven by the food intake and physical activity and sleep. And these behavior could be altered by the menstrual cycle. So we need data that look at all of these factors and compare them. And so this is one of the collaboration I have that's ongoing with the digital epidemiology lab at EPFL, and they have been leading the food and use study, which is a study where participants were continuous glucose monitoring for 2 weeks and take pictures of everything they eat, so that we have a very detailed food diary of what individual eats. And so when they started to design that study, I was visiting a postdoc in the lab, and so I was like, oh, maybe instead of following individuals for 2 weeks, we could have a sub cohort to menstruating women cohorts that, instead of tracking for just 2 weeks, would be tracking for a whole 4 weeks. And so that's kind of spans a full menstrual cycle for most individuals. And so this is the kind of data that we have. So for this is here only 5 days of data. So the blue rectangles is when people sleep, and the orange line is their glucose level. And here, this is the composition of what they eat, right? So from the pictures that they took, there is an algorithm that do image recognition, and then that is validated by some annotators to have then that translated to a quantity of nutrients. And so here, the idea would be to fit a model to be able to predict the curve from these nutrients, and then see whether across all the individuals, the menstruating women that have been enrolled in that additional cohort, whether there is any systematic bias at some phases of the menstrual cycle, either in the glycemic response, and that would indicate some direct effect, or through the modification of the behavior in terms of food or physical activity. So this is for some of the future that I'm excited to do. And of course, these participants have been tracking their cycle, so we know at which phase of the menstrual cycle they are. So with this, I'm going to thank you, my collaborators, the funders too. So the vaginal microbiota work has been funded by the Gates Foundation, some of the earlier work by the Swiss National Foundation and the Stanford Clinical Data Science Fellowship. And there are lots of collaborators I need to thank here. And so with this, I'd be happy to take any question if there are any. Thank you so much.
Yes, definitely. Thanks a lot, Laura. That leaves us with plenty of time for questions. Tel, please go ahead.
Oh, thanks. Really appreciate that talk, Dr. Symul. Really enjoyed it. You know, I'm curious, you mentioned that using topic modeling compared to other, or at least one other clustering approach, that in this domain, that you had found a better predictive performance. And I'm curious whether you've evaluated that in other domains, or whether you've looked at any sort of theoretical advantages or even guarantees in that comparison.
Yeah, that's a really good question. So I think one of the reasons that it works so well in the vaginal microbiota is that it's less diverse microbiota. But so what I would expect is that in the gut microbiota, more topics would be needed to be able to represent the bigger diversity that there is in the gut. And then I also, so I'm not an expert of the gut microbiota. My understanding is that it's a little more stable than the vaginal microbiota. And so there is maybe less of a need to be able to predict transitions. But I could imagine that, you know, in studies after people have had antibiotics, for example, or in studies where there is an intervention with probiotics, then it would be useful to understand if there are some subcommunities that are, you know, that react better or worse to those interventions. Does that answer your question? I think there was a second part, but I can't remember.
No, that's really great context related to sort of this domain of microbiota. I guess I was more thinking about it methodologically. It sounds like maybe you're talking about a function of K or perhaps class balance, where there would be scenarios where topic modeling or topic alignment would be the more appropriate, or perhaps more useful method. Am I hearing that right?
Yeah. So, all right. So one of the reasons why, in this case, for example, topic model does a lot better also is because transitions happen. So they might be stochastic, but also if you have a good bug that has, and you see something that starts to grow, then topic model is going to capture that signal, right? Like it's going to know that even if it's only a small piece of something else that's growing, then there is a possibility that that thing will continue to grow and eventually take over. So this is because so, and it's not very fair necessarily to compare, the dimensions of clusters versus the dimension of topics. You can say you have less topic, but it's still a mixed membership model. So the dimensions are not really comparable, but you still, from an interpretation perspective, you still are dealing with less categories that you need to remember and interpret as humans than you would with topics. And so this is where the benefit is. But again, so we did it step by step here, where first looking at what is the optimal number of topic, and then not necessarily looking into that then for the predictive performances of the transitions.
No, that's great. Thank you. Just thinking about potential translation of the work, it is a little bit trickier concept to explain to non-experts, and so
yeah,
great to hear that you're sort of thinking about that for the future.
Yeah, no, I agree. Not always easy to explain, but it works. Like, yeah. Yeah.
I had a follow-up question about so with the gut, I was curious kind of like computationally how this would apply to like a much more complex ecosystem. And then also like in the gut, like sometimes they talk about enterotypes, and there are not very many. Like some of those papers, at least the initial papers, were like, about three main kind of enterotypes, like clustering groups. And so I think the membership might be like more diverse, but there's lots of things in those enterotypes, but they're kind of dominated by a couple different taxa. Like, how well do you think this method would apply to the gut? Do you think you would just need, like you said, that maybe you would need lots of topics, but like computationally, do you think it would work well, or like in terms of interpretation and whatnot, do you think it would apply well, or have you thought about extending it to or like applying it to the gut?
Yeah, so computationally, it actually is not too heavy. Also, it's a Bayesian model, but I mostly, there is GIP sampling or variational, like EM, and so that actually runs pretty fast and gives results that are pretty similar. And also, I guess there would be also a lot of potential looking at the posterior distributions, and it was already like so much of an improvement that I haven't even started to look at that. But I think it probably would be a little more computationally extensive on the gut, and maybe there would be more precautions to take because the data is less sparse than in the vaginal microbiota. And so, for example, the prior distribution of the parameters that we used when we fit this model almost don't matter. We can set the Dirichlet parameter to approximate whatever we want, and we always have very similar results. I would expect this could be different in the gut, and that there could be more precautions that have to be taken with respect to the prior values of those parameters. That's what I expect. Yeah.
I have one other question that I think is quick. So with Alta, were you applying that cross-sectionally to like different times, or is there like some longitudinal aspect of the method?
So no, in this case, it was all cross-sectional, and I think there is also a lot of, I think that's a really good question. And I'm thinking a little bit about how to integrate the longitudinal aspect and correlation, right? Like just data are more correlated within individuals that could be useful. Yeah. Mm-hmm.
Interesting. Thanks.
Thank you.
I have a couple of questions about, you mentioned multi-omic alignment, and I found that really, an exciting next step for your method. So I guess, so two questions is, can you talk a little bit about what do you mean by, these different omics types that you're currently thinking about for the applications? And I guess my question was more along the lines of what Maggie was asking, especially if the scale of the dimensionality and the scale of the data increases massively with various types of omics data, how does your method, you think, copes with this increasing scale? And also the other question that I have is that, maybe I'm not understanding this correctly, but is there some notion of doing alignment across patients also, or is this alignment with data within a patient? So if you could clarify that, that'll be great.
Yes, yeah. So I think the first and last questions are somewhat related, if I understand what you were asking. So in terms of alignment across omics, so there are a lot of approaches there to be taken, but one where I think topic alignment could be almost directly applied is in aligning topics that are fitted on metagenomic data and metatranscriptomic data. And so there one could have a very naive approach of just like really looking at the alignment and seeing like what is similar and what is different. But this is also where I think that adding constraints on the topic. So instead of doing topic alignment, so metagenomic and metatranscriptomic, if for example we quantify the counts at the gene level, then your expectation is that for a gene to be transcribed, then it has to be there. And of course that might not always be the case, just because of the sequencing depth that is different. And so you could have a transcript where you have no reads, but no genes, but that should be rare, and you can still model this, right? Taking a multinomial from your metagenomics. So one of the things is just simply, and I'm going to try to go back a few slides, see if I can show that. Well, actually, this could help. But so if you remember, like this dual diagram between topic composition and sample composition. So here, this is at 2 different resolution, right? But you have the sample composition for one model and the sample composition for the other one. And so nothing would prevent to have 2 different modalities, right? Here for like metagenomic, for example, and metatranscriptomic, and look what is different or the same. But in addition, when you look at metatranscriptomic and metagenomic, you could also look at the beta composition and do the alignment on the beta. So that would be an ideal case to just apply alignment to this kind of data, which I've partly done, but it was more like exploratory, and it gave interesting results. But I think that there will be even more interesting results once we also explicitly model the fact that a transcript can only exist if a gene is present. So yeah. So that was one of the first questions, and then I guess one question was on the scale of the data and how that would scale. And yes, well, so there in the field, I think of multi-omic alignment or multi-view alignment, depending, or multi-table, like depending on the field, everyone calls it a bit differently. But there is a little bit division of early fusion versus late fusion, and how much to reduce on one dimension to be able to cope with the amount of data. And so I've actually had some luck like applying that to, so in one of the last slides, I was, I think, looking at that here. So one of the outcome was whether a participant had BV or not. And we had indeed like different omics, but also a lot of clinical data, right, and behavioral data. And so just by reducing the microbiota composition to using topics, then we had several blocks, and we were still able to find very strong associations between that. And computationally, it's not so bad, right? It does work. It was actually okay. Yeah.
Questions?
I had a question.
Yes, please.
So in the slide, the one before the last slide, while you were talking about future work, you said you talked about the effects of menstrual cycle on the glucose regulation, and you wanted to. So yeah, like you have sleep and physical activity, food intake, lots of data. Do you have any vision? How would you incorporate all these different data sources for glucose regulation? We already talked a little bit about this, but when I see the slide here, I think it made more sense to me now.
Yes, yeah. So currently the idea, but it's still something I'm exploring, right, in terms of what would be the right approach, and exactly along the line of what we talked earlier. So one naive approach is simply to consider an additive model where each nutrient has a response, and you model that with a few parameters, and then you. So one of the key aspect also to this data is that even though it's really high resolution, it's also self-reported, right? So individuals, they took pictures. So there are a lot of sources of uncertainty and imprecision in this data. So first of all, the timing, right? People may have uploaded their picture a little earlier or a little later, but on the time scale we're talking, it could actually matter. And then from pictures, it might not be, we know that it's actually a good reflection of like the amount, right? So there is uncertainty on the timing and on the importance here. And so one of the approaches I've been considering is a pretty simple Bayesian model where you also model the uncertainty and both the amplitude of the quantity and the timing of the quantity, and you put priors that are mostly like exponential priors, for example, like centered on 0 on 1, and then see if you can recover those parameters for individuals. So yeah.
Thank you very much. And one last thing, how many individuals' data is being collected? Do you have any?
Yeah, so the cohort is 100 menstruating individuals.
And each of them for 4 weeks.
4 weeks, yes.
Okay, thank you very much.
Yeah. And maybe just to follow up on this, and this is something of a bridge to sort of a Chalk Talk discussion, but you showed really lovely visualizations, by the way. You showed great depth and granularity of data from some very sophisticated studies, and certainly partnership with folks like the digital epidemiology lab you mentioned to be able to analyze some of the data that they are collecting can be one kind of pathway to success. I'm curious if you thought about sort of, what the study would look like that you would want to launch, if in your new faculty role.
Yeah, so with this, I would very much look forward to meet more individuals at the medical school. There are always like different ways of collecting data, and depending on the questions, right, that want to be asked. But one of the things, and I mean, this study will also serve a little bit as a validation for tracking menstrual cycle with an app, and see if that works, and see if that is deployable in large scale. So another topic that I'm pretty interested in is in the PMDD diagnostics. So PMDD stands for premenstrual dysphoric disorder, and so it's in the DSM-5. And so first of all, there's 24 symptoms, right, that make up that diagnostic. And it's already been shown that there are different temporal trajectories. And again, people have used clustering approaches to look at those temporal trajectories. It's very likely not clusters, it's more likely a continuum. But then not just in the temporal trajectories, but in the type of symptoms that people experience, we can expect that not everyone has the same set of symptoms. And so there, even as a non-clinician person, there are already a lot of apps that collect data that are very close to the symptom definitions in the DSM-5. And so one of the things that I could do even independently would be to partner with these apps, as I've already done for having retrospective data, but to prospectively collect data. And then have like follow people over long periods of time and include additional questions that could drive changes in symptoms or about the lifestyles. But yeah.
That's great. Thank you. I think we'll enjoy the conversation tomorrow in the Chalk Talk.
Yeah, looking forward to it.
All right. Any final questions? Okay. Going once, twice. Okay. All right. Dr. Symul, thank you so much for the talk. We enjoyed it, and we're looking forward to all the other interactions with you. See you.
Thanks. Bye-bye.
See you, everyone.